continuation of cancer topic. Cancer cells escape the confines of tissues. Normal cells stop growing when they contact other cells, eventually forming a layer of well-ordered cells. Cancer cells are less adherent, forming a three-dimensional cluster of cells, a focus that can be recognized under a microscope. This loss of confinement to tissue structure can be modeled in vitro. Cultured mouse fibroblasts called 3T3 cells normally stop growing when they contact other cells, eventually forming a multi-layer of well-ordered cells that have stopped proliferating and are in the quiescent G0 phase of the cell cycle. When DNA from human bladder, human cancer cells is transferred and transfected into cultured 3Tc cells. About one cell in a million incorporates a particular segment of the exogenous DNA that causes a phenotypic change. The progeny of the affected cells are not rounded and less adherent to one another and to the culture dish than are the normal surrounding cells. Cells that do not cease division when they contact other cells are said to be no longer contact inhibited and to have undergone oncogenic transformation. Recent work has implicated adhesion molecules such as E-cadherin, cell polarity factors, and adhesion molecules such as E-cadherin, cell polarity factors, actin, cytoskeleton regulators, and the Hippo pathway in the mediating cell cycle arrest when cell-cell contacts are established. However, the exact mechanisms whereby this occurs and how these pathways are disrupted in cancer remain to be worked out. Then next topic, tumors are heterogeneous organs that are sculpted by the environment. Not all tumors are made up of uniform cells, even if they originated from a single initiating cell. In some types of tumor cells, for example, only certain tumor cells called cancer stem cells are capable of seeding a new tumor. Then within this tumor, some cells cease dividing while others can continue cancerous growth. The latter, of course, are the most dangerous and the most important to destroy with anti-cancer treatments. Cancer stem cells are thought to give rise to some cells with high replicative capabilities and others with more limited replicative potential. The origins of these cancer stem cells are not clear. In some cases, a normal tissue stem cell may give rise to the cancer stem cell. In others, de-differentiation of terminally differentiated cells to form progenitor cells may give rise to cancer stem cells. So before moving on to that, let's see a picture which depicts a scanning electron micrograph which reveal the organizational and morphological differences between normal and transformed 3T3 cells. Cultured mouse fibroblasts called 3T3 cells are normally elongated and are aligned and closely packed in an orderly fashion. 3T3 cells transformed by an oncogene encoded by Rao sarcoma virus are rounded and covered with small hair-like processes and bulbous projections, and the transformed cells have lost the side-by-side organization of the normal cells and grow one atop the other. These transformed cells have many of the same properties as malignant cells. Similar changes are seen in cells infected with DNA from human cancers containing the RAS-D oncogene. The immediate environment of a tumor, the tumor microenvironment, contributes to the heterogeneity of cells within the tumor, influencing the behavior of cancer stem cells and the tumor cells in general. The immediate environment of a tumor, the tumor microenvironment, contributes to the heterogeneity of cells within the tumor, influencing the behavior of the cancer stem cells and tumor cells in general. Some neighboring cells may be more conducive to tumor growth than others. The importance of tumor microenvironment extends to one of the most common environmental influences on a tumor cell, inflammatory cells. It is now widely accepted that cells of the immune system interact with the tumor CD8 plus cytotoxic T lymphocytes and natural killer cells surround and often migrate into the tumor, where they are thought to inhibit tumor formation. Mice deficits in this and other components of immune system are more prone to carcinogen-induced tumors than normal mice. This finding led to the idea that immune system eliminates cancer cells. How cancer cells escape this immune surveillance is a critical question that remains to be addressed. More and more evidence is mounted that immune system cells can also have tumorogenic properties. It has been known for a long time that cancers frequently arise at sites of injury or chronic infection. It is estimated that up to 20% of cancers are linked to chronic infection. For example, persistent Helicobacter pylori infection is associated with gastric cancer, and Crohn's disease, an autoimmune disease that affects the intestine, is associated with colon cancer. Infected with hepatitis B or C virus increases the risk of a form of liver cancer, hepatocellular carcinoma. Immune system cells migrate to sites of injury or infection and produce growth factors, thereby stimulating tumor cell proliferation. They also produce factors to induce the growth of blood vessels, which, as we will discuss next, is an essential part of tumor growth and dissemination to distant sites. Then, tumor growth requires formation of new blood cells. Tumors must recruit new blood vessels in order to grow to a large size. In the absence of a blood supply, a tumor can grow into a mass of about 10 raised to 6 cells, roughly a sphere 2 mm in diameter. At this point, division of cells on the outside of tumor mass is balanced by death of cells in the center from an inadequate supply of nutrients. Such tumors, unless they secrete hormones, cause few problems. However, most tumors induce the formation of new blood vessels that invade the tumor and nourish it, a process called angiogenesis. The complex process requires several discrete steps: degradation of the basement membrane that surrounds a nearby capillary, migration of endothelial cells lining the capillary into the tumor, division of the endothelial cells, and formation of a new basement membrane around the newly elongated capillary. Many tumors produce growth factors that stimulate angiogenesis. Other tumors somehow induce surrounding normal cells to synthesize and secrete such factors. Basic fibroblast growth factor, or beta FGF, transforming growth factor, alpha, TGF alpha, and vascular endothelial growth factor, VEGF, which are secreted by many tumors, all have angiogenic properties. New blood vessels allow the tumor to increase in size and thus increase the probability that additional harmful mutations will occur. The presence of an adjacent blood vessel also facilitates the process of metastasis. The VEGF receptors, which are tyrosine kinases, regulate several aspects of blood vessel growth, such as endothelial cell survival and growth, endothelial cell migration, and cell wall permeability. VEGF expression can be induced by oncogenes and by hypoxia, defined as a partial pressure of oxygen of less than 7 mmHg. The hypoxia signal is mediated by hypoxia-inducible factor 1, HIF1, a transcription factor that is activated in low oxygen conditions and which binds to and induces transcription of the VEGF gene. And about 30 other genes, many of which can affect the probability of tumor growth. HIF1 activity is controlled by an oxygen sensor composed of a prolyl hydroxylase that is active at normal O2 levels but inactive when deprived of O2. Hydroxylation of HIF1 causes ubiquitinalization and degradation of the transcription factor, a process that is blocked when O2 is low. Compounds that inhibit angiogenesis have excited much interest as potential therapeutic agents, but their success in the clinic has thus far been limited. Then invasion and metastasis are late stages of tumorigenesis. Tumors arise with greater frequency, especially in older individuals, but most pose little risk to their host because they are small and localized. We call such tumors benign. They are of pose little risk to their host because they are small and localized. And an example is a benign skin tumor. The cells composing benign tumors closely resemble and many may function like normal cells. The cell adhesion molecules that hold tissues together keep benign tumor cells like normal cells localized to the tissues where they originate. A fibrous capsule usually delineates the extent of benign tumor and makes it an easy target for a surgeon. Benign tumors become serious medical problems only if their sheer bulk interferes with normal functions or if it secretes excess amounts of biologically active substances such as hormones. For example, acromegaly, the overgrowth of head, hands, and feet, can occur when a benign pituitary tumor causes overproduction of growth hormones. In contrast to benign tumor cells, malignant tumor cells are able to invade nearby tissues, spreading and seeding additional tumors while the cells continue to proliferate. This ability is a major characteristic that differentiates malignant tumors from benign ones. So, malignant tumors, such as those in the ovary or breast, remain localized and encapsulated at least for a time. When these tumors progress, however, the cells invade surrounding tissues and undergo metastasis. Normal cells are restricted to their place in an organ or tissue by cell-cell adhesion and by physical barriers such as the basement membrane, which underlie layers of epithelial cells and also surrounds the endothelial cells of blood vessels. In contrast, cancer cells have acquired the ability to penetrate basement membranes using a cell protrusion called an invadopodium and to migrate to distant sites in the body. So here is a picture which shows gross and microscopic views of a tumor invading normal liver tissue. So the gross morphology of human liver in which a metastatic lung tumor is growing. The white protrusions in the surface of the liver are tumor masses. A light micrograph of a section of the tumor showing areas of small dark staining tumor cells invading a region of large light staining normal liver cells. Tumor cells are there, we can see, and normal cells are there. A developmental process known as the epithelial to mesenchymal transition is thought to play a crucial role during the process of metastasis in some cancers. During normal development, the conversion of epithelial cells into mesenchymal cell is a step in the formation of some organs and tissues. An EMT requires distinct changes in patterns of gene expression and results in fundamental changes in cell morphology, such as loss of cell-cell adhesion, loss of cell polarity, and the acquisition of migratory and invasive properties. During metastasis, the EMT regulatory pathways are thought to be activated at the invasive front of the tumor, producing single migratory cells. At the heart of the EMT are two transcription factors, snail and twist. These transcription factors promote expression of genes involved in cell migration, trigger downregulation of cell adhesion factors like E-cadherin, and increase the production of proteases that digest the basement membrane, thus allowing its penetration by the tumor cells. For example, many tumor cells secrete a protein like plasmogen activator that converts the serum protein plasminogen to the active protease plasmin. Importantly, expression of many important drivers of the EMT, such as SNAIL-1 and SNAIL-2, has been shown to correlate with disease relapse and decreased patient survival in many cancers, including breast, colon, and ovarian cancer. The occurrence of the EMT predicts a poor clinical outcome. As the basement membrane disintegrates, some tumor cells enter the bloodstream, but fewer than 1 in 10,000 cells that escape the primary tumor survive to colonize another tissue and form a secondary metastatic tumor. Much of the preventative medicine is currently focused on developing methods to identify the rare tumor cells that circulate in the bloodstream. The ability to capture the circulating tumor cells would not only provide a powerful and non invasive tool for the early detection of cancer, but their analysis could provide insights into the nature of the disease and inform treatment. In order to produce metastasis, tumor cells must not only enter the bloodstream but adhere to the lining of the blood vessel in a new location and migrate through it. In this picture, metastasis is represented. First step in metastasis, using breast carcinoma cells as an example. Cancer cells leave the main tumor and attack the basement membrane using extracellular matrix or ECM fibers to reach the blood vessels. The cancer cells can be attracted by signals such as epidermal growth factor or EGF, which can be secreted by macrophages. At the blood vessels, they penetrate the layer of endothelial cells that forms the vessel walls and enter the bloodstream. Then carcinoma cells penetrate the extracellular matrix and blood vessels wall by extending invadopodia, which produce matrix metalloproteases and other proteases to open up a path. So primary tumor is there, carcinoma cell is there, basement membrane is there, degradation of basement membrane by invadopodia. Then carcinoma cell migrating on ECM fiber can be seen. Then EGF secreted from macrophages and diffused from blood vessels. Along with that, degradation of basement membrane by invadopodia is there. Migration on ECM, invasion through ECM, coffulin and VASP are seen. Actin, invadopodium and ECM, etc. can be seen in this picture. And the underlying tissue in a process is called extravasation. In order to seed a metastasis at a distant site, the tumor cells must not only disseminate but also adapt to a foreign tissue environment. At least initially, metastatic tumor cells may not be well adapted to their new environment, but they are thought to evolve to survive and thrive in a foreign context. Little is known about the molecular pathways that facilitate this adaptation, but evidence suggests that some environments are more conducive to cancer cell colonization than others. Because metastasis is the most common reason for morbidity associated with cancer, much effort is being put into understanding which tumors will become metastatic and how metastasis occurs. Traditionally, the properties of tumor and normal cells have been assessed using microscopic tools, and the prognosis of many tumors could be determined within certain limits from their histology. However, the appearance of cells alone has limited information content, and better ways to discern the properties of cells are desirable, both to understand tumorigenesis and to arrive at meaningful and accurate decisions about prognosis and therapy. The advent of methods to assess a tumor's patterns of RNA, protein, lipid, and metabolite production is allowing for a more detailed examination of tumor properties. Not surprisingly, primary tumors are often distinguishable from metastatic tumors by the RNAs and proteins that they produce. Analysis of global patterns of gene expression are now routinely used to predict patient outcomes and to determine the best course of treatment for many other cancers, and they will soon become the standard in determining treatment options. So some major key concepts of this section are involved in this topic, like how tumor cells differ from normal cells. The genomes of most cancer cells undergo dramatic alterations ranging from point mutations to deletions and amplifications to whole chromosome gains and losses. These changes in genetic makeup affect virtually all cellular functions. Then uncontrolled proliferation and escape from the confines of the tissues of origin are two universal traits of cancer cells. Then tumors are complex organs composed of different cell types that interact with their environment. to obtain a maximal growth advantage. Then both primary and secondary tumors require angiogenesis, the formation of new blood vessels in order to grow to a large mass. Then cancer cells sometimes invade surrounding tissues, often breaking through the basement membrane that define the boundaries of tissues and spreading through the body to establish secondary areas of growth, a process called metastasis. Then metastatic tumor cells acquire migratory properties in a process called epithelial to mesenchymal transition. Then next topic, the origins and development of cancer. Tumors arise from single cells that acquire the ability to proliferate when the neighbors cannot. A series of evolutionary steps then follows by which cancer cells gain the means to escape the confines of tissues they originate in, to survive in the circulatory systems of the body, and finally to colonize distant sites. In this section, we examine the process of tumorigenesis. We first ask how carcinogens can induce tumorigenesis. We then induce a hypothesis known as multi-hit model of cancer and explains not only the multi-step path of the disease but also the fact that cancer is largely a disease of old age. We conclude this section by discussing cell-based and mouse models that have been instrumental in elucidating the molecular basis of tumorigenesis. Then carcinogens induce cancer by damaging DNA. The ability of cancer carcinogens, chemical carcinogens, to induce cancer results from the DNA damage they cause as well as the errors introduced into DNA during the cell's efforts to repair that damage. Thus, carcinogens are also mutagens. The strongest evidence that carcinogens act through mutagenesis comes from the observations that cellular DNA altered by the exposure of cells to carcinogens can change cultured cells or cells implanted in mice into fast-growing cancer-like cells. The mutagenic effect of a carcinogen is roughly proportional to its ability to transform cells and induce cancer in human animal models. Although substances identified as chemical carcinogens have a broad range of chemical structures with no obvious unifying features, they can be classified into general categories like direct acting carcinogens, of which there are only a few, are mainly reactive electrophiles, compounds that seek out and react with electron-rich centers in other compounds. By chemically reacting with nitrogen and oxygen atoms in DNA, these compounds can modify bases in DNA so as to distort the normal patterns of base pairing. If the modified nucleotides are not repaired, they allow an incorrect nucleotide to be incorporated during replication. This class of carcinogens include ethylamidine sulfonate or EMS, dimethyl sulfate or DMS, and nitrogen mustards. In contrast, indirect acting carcinogens are generally unreactive, often water insoluble compounds that can act as potent cancer inducers only after the introduction of electrophilic centers. In animals, cytochrome P450 enzymes are located in the endoplasmic reticulum of most cells and at especially high levels in liver cells. P450 enzymes normally function to add electrophilic centers such as OH groups to nonpolar foreign chemicals such as certain insecticides and therapeutic drugs in order to solubilize them so that they can be excreted from the body. However, P450 enzymes can also turn otherwise harmless chemicals into carcinogens. Indeed, most chemical carcinogens have little mutagenic effect until they have been modified by cellular enzymes. Then, some carcinogens have been linked to specific cancers. In the earliest days of cancer awareness, it becomes clear that at least some cancers are due to environmental poisons. In 1775, for example, it was reported that the exposure of chimney sweeps to soot causes scrotal cancer, and in 1791, the use of snuff or tobacco was reported to be associated with nasal cancer. Environmental chemicals were originally associated with cancer through experimental studies in animals. The classic experiment is to repeatedly paint a test substance on the back of a mouse and look for development of local or systemic tumors in the animal. Such assays led to the purification of a pure chemical carcinogen, benzopyrene, from coal tar in 1933. The role of radiation in damaging chromosomes was first demonstrated in the 1920s using gamma irradiated Drosophila. Later, the ability of radiation to cause human cancers, especially leukemia, was dramatically shown by the increased rate of leukemia among survivors of the atomic bombs dropped in World War II, ionizing radiation, and more recently by the increase in melanoma or skin cancer in individuals exposed to too much sunlight or ultraviolet radiation. Although chemical carcinogens are believed to be risk factors for many human cancers, a direct link to specific cancers have been established in only a few cases, the most important being lung cancer and other cancers of the larynx, pharynx, stomach, liver, pancreas, bladder, cervix, and more that are associated with smoking. Epidemiological studies first indicated that cigarette smoking was the major cause of lung cancer, but the reason was unclear until the discovery that about 60% of human lung cancer contain inactivating mutations in the p53 gene, which we will be soon see in a major tumor suppressor gene. The chemical benzoapyrene found in cigarette smoke as well as in coal tar undergoes metabolic activation in lungs to form a potent mutagen that mainly causes conversion of guanine to thiamine bases, a transversal mutation. So a picture, another graph is there which shows chemical carcinogenesis by tobacco smoke, which shows cigarette smoking provides a clear example of a deadly form of chemical carcinogenesis. The rates of lung cancer follow the rates of smoking, which with about a 30-year lag. Women began to smoke in large numbers starting in the 1960s, and starting in the 1990s, lung cancer passed breast cancer as leading cause of women's cancer deaths. At the same time, a gradual decrease in smoking rates among men started in the 1960s has been reflected as a decrease in their lung cancer rates. Then another picture which shows enzymatic processing of benzoapyrene to a more potent mutagen and carcinogen, like less carcinogenic metabolite is the benzoapyrene that will transform to benzoapyrene 7,8 epoxide, then benzoapyrene 7,8 diol, then benzoapyrene 7,8 diol 9,10 epoxide. After that, guanine has modified by the adduct at position N2, then plus trans anti B PN square DG is there, then mutation cancer DNA repair. Liver enzymes, particularly P45 450 enzymes, modify benzoapyrene in a series of reactions producing 7,8 diol 9,10 epoxide, a highly potent mutagenic species that reacts with DNA primarily at the N2 atom of a guanine base. The resulting adduct plus trans anti B APN square DG causes polymerase to insert an A rather than a C opposite the modified G base. Next time the DNA is replicated, a T will be inserted opposite to A and the mutation will be complete. Horizontal arrows indicate alterations toward greater potency, while vertical arrows indicate the changes in the direction of reduced toxicity. The large O symbol represents the rest of the multi-ring structure shown in the complete benzoapyrene molecule at the left. Causes conversion of guanine to thymine bases a transverse mutation. Then, when applied to cultured bronchial epithelial cells, activated benzo alpha pyrene induces many mutations, including inactivating mutations at codons 175, 248, and 273 of the p53 gene. The same positions all within the protein's DNA binding domain are major mutational hotspots in human lung cancer. In fact, the nature of the mutations in p53 and other cancer related genes found in tumor cells gives us cues as to the origin of cancer. Then, the same positions all within the protein's DNA binding domain are major mutational hotspots in human lung cancer. In fact, the nature of the mutations in P53 and other cancer-related genes found in tumor cells give us clues as to the origin of the cancer. The G to T strand conversions caused by benzoapyrene, for example, are present in the P53 genes of about one-third of smokers' lung tumors. This type of mutation is relatively rare among the P53 mutations found in other types of tumors. The carcinogen leaves its footprint. Thus, there is a strong correlation between one defined chemical carcinogen in cigarette smoke and human cancer. It is likely that other chemicals in cigarette smoke induce mutations in other genes since it contains more than 60 carcinogens. Lung cancer is not the only major human cancer for which a clear-cut risk factor has been identified. Asbestos exposure is linked to mesothelioma, another type of lung cancer. Aflatoxin, a fungal metabolite found in moldy grains, induces liver cancer. Furthermore, cooking meat at high temperature causes chemical reactions. Cooking meat at high temperatures causes chemical reactions that form heterocyclic amines or HCAs, potent mutagens that cause colon and breast carcinomas in animal models. Hard evidence concerning dietary and environmental risk factors that would help us avoid other common cancers like breast, colon, and prostate cancer and leukemias is, however, generally lacking. Then, the multi-hit model can explain the progress of cancer. As we have seen, mutations cause cancer. However, luckily for us, multiple mutations are usually required to convert a normal body cell into a malignant one. According to this multi-hit model, cancers arise by a process of evolutionary or survival of the fittest. Clonal selection, not unlike the selection of individual animals in a large population. Here in the scenario, which may or may not apply to all cancers, a mutation in one cell gives it a slight growth advantage. One of its progeny cells then undergone a second mutation that allows its descendants to grow more uncontrollably and form a small benign tumor. A third mutation in a cell within this tumor allows it to overgrow the others and overcome constraints imposed by the tumor microenvironment with its progeny from form a mass of cells, each of which has these three genetic changes. An additional mutation in one of these cells allows its progeny to escape into bloodstream and establish daughter colonies at other sites, the hallmark of metastatic cancer. This model makes two easily testable predictions. First, a cell in a given tumor should have at least some genetic alterations in common. Systematic analysis of cells from individual human tumors supports the prediction that all the cells in a tumor are derived from a single progenitor. Second, cancer incidences should increase with age because it can take decades for the required multiple mutations to occur. So here we can see a graph which shows the incidence of human cancers increase as a function of age. The marked increase in the incidence of cancer with age is consistent with the multi-hit model of cancer induction. Note that logarithm of annual incidence is plotted versus the logarithm of age. Then we can see the picture. Prostate, stomach, skin, rectum, pancreas, and esophagus cancers are involved. Annual incidence per 100,000 males. Assuming that the rate of mutations is roughly constant during a lifetime, the incidence of most types of cancer would be independent of age if only one mutation were required to convert a normal cell into a malignant one. As the data in the figure show, the incidence of many types of human cancer increase drastically with age. In fact, current estimates suggest that 5 to 6 hits or mutations must accumulate as the most dangerous cancer cells emerge. Then, more direct evidence that multiple mutations are required for tumor induction comes from experiments with transgenic mice, which have shown that a variety of combinations of oncogenes can cooperate in causing cancer. For example, mice have been made that carry either the mutant RASV12 dominant oncogene, one version of RASD, or the MYC proto-oncogene, in each case under the control of a mammary cell specific promoter or enhanced from a retrovirus. This promoter is induced by endogenous hormone levels and tissue specific regulators, leading to overexpression of MYC or RASV12 in breast tissue. The MYC protein is a transcription factor that induces expression of many genes required for the transition from the G1 to the S phase of the cell cycle. Heightened transcription of MYC in this mice mimics previously identified oncogenic mutations that increase MYC transcription, converting the proto-oncogene into an oncogene. By itself, the MYC transgene causes tumors only after 100 days, and then them in only a few mice. Clearly, only a minute fraction of the mammary cells that overproduce the MYC protein actually become malignant. Then production of the mutant RASV12 protein alone causes tumors earlier, but still slowly and with about 50% efficiency over 150 days. When the MYC and RASV12 overexpressing transgenics are crossed, however, all mammary cells in the offspring overproduce both MYC and RASV12. Tumors arise much more rapidly, and all animals succumb to cancer. Such experiments emphasize the synergistic effects of multiple oncogenes. They also suggest that the long latency of tumor formation seen even in the double transgenic mice is due to the need to acquire still more mutations. Then a graph is here. The kinetics of tumor appearance in female mice carrying either one or two oncogenic transgenes shows the cooperative nature of multiple mutations in cancer induction. One of these transgenes are driven by the mouse mammary tumor virus, MMTV virus, breast specific promoter. The hormonal stimulation associated with pregnancy activates the MMTV promoter, and hence the overexpression of the transgenes in mammary tissue. The graph shows the time course of tumorigenesis in mice carrying either MYC or RASV12 transgenes, as well as in the progeny of a cross of MYC carriers with RASV12 carriers which contain both transgenes. The results clearly demonstrate the cooperative effects of multiple mutations in cancer inductions. Then, successive oncogenic mutations can be traced to colon cancers. Studies on colon cancer provide the most compelling evidence to date for the multi-hit model of cancer induction. Surgeons can obtain fairly pure examples and samples of many human cancers, but since the tumor is observed at only one time, its exact stage of progression cannot be easily determined. An exception is colon cancer, which evolves through distinct, well characterized morphological stages. Its intermediate stages like polyps, benign adenomas, and sarcomas can be isolated by a surgeon, allowing mutations that occur in each of the stages to be identified. Numerous studies have shown that colon cancer arises from a series of mutations that commonly occurs in a well-defined order, providing strong support for the multi-hit model. Insight into the progression of colon cancer first came from the study of inherited predispositions to colon cancer, such as familial adenomatous polyposis. Mutations in the WNT signaling pathways have been identified in many of these syndromes, and it is now believed that deregulation of WNT signaling results in formation of polyps, pancreatitis growths, and on the inside of the colon wall, not only in people with inherited polyposis syndromes, but also in people afflicted with sporadic or non-inherited forms of colon cancer. The APC or adenomatous polyposis coli protein is a negative regulator of WNT signaling, which promotes cell cycle entry by activating expression of the MYC gene. The absence of functional APC protein thus leads to inappropriate production of MYC, and cells homozygous for APC mutations proliferate at a rate higher than normal and form polyps. Loss of function mutations in the APC gene are the most frequent mutations found in early stages of colon cancer. Most of the cells in a polyp contain some one or two mutations in the APC gene that result in its loss or inactivation, indicating that they are clones of the cell in which the original mutation occurred. Thus, APC is a tumor suppressor gene, and both alleles of the APC gene must carry an inactivating mutation for polyps to form, because cells with one wild type APC allele increase enough APC protein to function normally. If one of the cells in a polyp undergoes another mutation, this time an activating mutation of the RAS gene, its progeny divide in an even more uncontrollable fashion, forming a larger adenoma. Inactivation of the p53 gene follows and results in the gradual loss of normal regulation and the consequent formation of a malignant carcinoma. The p53 protein is a tumor suppressor that halts progression through the cell cycle in response to DNA damage and other stresses. While the three hits listed here are certainly crucial parts of the picture, there are likely to be an additional contributing genetic events. Not every colon cancer, however, acquires all the later mutations or acquires them in the order. These different combinations of mutations may result in the same phenotype. DNA from different human colon carcinomas generally contains mutations in all three genes mentioned here. Loss of function mutations in the tumor suppressor APC and p53, and the activating gain of function mutations in the oncogene KRAS, one of the RAS family of genes, establishing that multiple mutations in the same cells are needed for the cancer to form. Some of these mutations appear to confer growth advantages at an early stage of tumor development, whereas other mutations promote the later stage, including invasion and metastasis, which are required for the malignant phenotype. The number of mutations needed for colon cancer progression may at first seem surprising and might seem to be an effective barrier to tumorigenesis. Our genomes, however, are under constant assault. Recent estimates indicate that sporadically arising polyps have about 11,000 genetic alterations in each cell, though very likely only a few of the alterations are relevant to oncogenesis. The genetic instability that is a hallmark of cancer cells promotes further tumor evolution, allowing for the accelerated creation of tumor cells with increased self-reliance and the ability to metastasize. Then colon carcinomas provide an excellent example of the multi-hit model of cancer. The degree to which this model applies to cancer generally is only now being learned, but it is clear that many types of cancer involve multiple mutations. Then cancer development can be studied in cultured cells and in animal models. In this picture, we can see a polyp. Small growth forms on the colon wall. This figure, the development of metastasis of human colorectal cancer and its genetic basis. A mutation in the APC tumor suppressor gene in a single epithelial cell causes the cell to divide. All the surrounding epithelial cells do not, forming a mass of localized benign tumor cells called a polyp. Subsequent mutations lead to the expression of constituently active RAS protein and loss of tumor suppressor gene p53. These mutations, together with additional genetic changes yet to be identified, generate a malignant cell. The cell continues to divide and its progeny invade the basement membrane that surrounds the tissue, but do not penetrate the basement membrane of capillaries, bottom left. Some tumor cells spread into blood vessels that will distribute them to other sites of the body. Additional mutations permit the tumor cells to exit from the blood vessels and proliferate at distant sites. So picture shows regions of a polyp, small growth forms on the colon wall, then normal colon cells are there. Loss of APC tumor suppressor gene, chromosome 5, activation of KRAS oncogene, chromosome 12, and loss of P53 tumor suppressor gene, chromosome 17, and other changes. And the benign precancerous tumor grows, a class 2 adenoma benign grows, a class 3 adenoma benign grows, then a malignant carcinoma develops. A cancer metastasis spread to other tissues will be happening, and tissue lumen of colon, polyp region, wall of colon, then some other regions like invasive tumor cells are there, normal colon epithelial cells, basement membrane, tumor cells invade blood vessels allowing metastasis to occur, and blood vessels can be seen. Some cells, however, escape this fate and become immortal, that is, they gain the ability to divide indefinitely. Immortalization is mediated by several kinds of mutations, including loss of function mutations in the P19ARF or P53 genes, which are regulators of the cell cycle and cell survival. These mutations allow cells to grow for an unlimited time in culture if they are periodically diluted and supplied with nutrients. Next topic we will be learning with the help of next video.